Hello, I'm Dr. Aruna Saman, and in this video, we will be discussing reproduction in higher plants. We will be mainly focusing on fertilization process in a flower. In my previous video, we discussed pre-fertilization changes occurring in a flower. We all know that the two major events occurring in a flower during sexual reproduction are the formation of haploid gametes and fusion of the gametes, which is called fertilization. A diploid zygote is formed as a result of fertilization, which then undergoes embryogenesis to form new offspring. The three distinct stages during sexual reproduction are pre-fertilization, fertilization, and post-fertilization. In angiospermic plants, a flower is meant for sexual reproduction. It's a modified stem. Here we can see major changes occurring in a flower during sexual reproduction. They are put under three stages: pre-fertilization, fertilization, and post-fertilization. We have already completed the discussion of the changes in the pre-fertilization stages. Let us discuss the second stage, fertilization. Fertilization is defined as the process of union of male and female gametes. After pollination, the pollen grain germinates on the stigma. A pollen tube is produced, which travels through the style. In the pollen tube, the two male gametes are produced. The pollen tube containing two male gametes is called male gametophyte. At the same time, the ovary in its ovule, megasporogenesis takes place and the functional megaspore is formed. The functional megaspore develops into an embryo sac or female gametophyte containing egg or female gamete. These changes come under pre-fertilization changes. They lead to the process of fertilization. In angiosperms, it is called double fertilization. Let us try to understand the process in detail. You can see here female gametophyte or embryo sac and male gametophyte, which is a pollen tube containing two male gametes. The pollen tube penetrates one of the synergids. Watery content of synergids are absorbed by pollen tube, due to which the tip of the pollen tube breaks. The male gametes are released in the embryo sac. One male gamete fuses with the egg to produce a diploid zygote. This is called syngamy. The other male gamete fuses with the polar nuclei to form a triploid cell called primary endosperm nucleus, PEN. This is triple fusion. Let us see an animated movement of the male gametes. We can see here the egg has received the male nucleus and it has become diploid zygote. The secondary nucleus or definitive nucleus has received the other male nucleus and it has become triploid primary endosperm nucleus. Here you will be able to notice the changes which occurred in the embryo sac after fertilization. See the egg or female gamete, which was haploid. It has now become a diploid zygote. The diploid secondary nucleus, it has become triploid primary endosperm nucleus. The synergids, they got disorganized. The antipodal cells, they also got degenerated after fertilization. Let us see the sequential events occurring during double fertilization and different scientific terms used to describe them. The pollen tube carries in its hollow part the two male gametes. This is called siphonogamy. It grows through style to reach the ovary chamber and to enter the ovule. Pollen tube enters ovule usually through the micropyle. This is called porogamy. Synergy directs the tube towards the egg and helps to break the tip of the pollen tube. Two male gametes are released. One male gamete fuses with the egg to produce a diploid zygote. This is called syngamy. The other male gamete fuses with the two polar nuclei to form a triploid cell called primary endosperm nucleus (PEN). This is called triple fusion. Since both the male gametes are utilized, the fertilization involves fusion at two places. This is called double fertilization. What are the main products of double fertilization? The syngamy or true fertilization. It is the fusion of male and female gametes results in the formation of diploid zygote. The zygote then undergoes embryogenesis to form an embryo. The embryo grows into a new plant and this is called a generative type of fertilization. Triple fusion is the other male gamete with diploid secondary nucleus cell. Resultant cell is triploid and it is called primary endosperm nucleus. This cell undergoes repeated divisions to produce a mass of cells called endosperm. It is rich in food. It is a type of fusion called vegetative fertilization. Some additional information we would consider here. We have already seen that pollen tube enters the ovule through the micropyle. It is termed as porogamy. But in some cases, it enters through chalaza. Then it is called chalazogamy. Whereas in some plants, it enters through integuments. Then it is called mesogamy. But the pollen tube penetrates the embryo sac always through its micropylar end. Let us see the significance of double fertilization. It is a characteristic feature of angiosperms. Hence, we can identify angiospermic plants using this feature. Both male gametes get utilized in this process. With syngamy, diploid condition gets restored and the sporophytic generation begins. The diploid zygote develops into an embryo which gives rise to new plant. The triploid PEN develops into nutritive endosperm tissue. It also helps avoid polyembryony. Plant invests its resources in the seed to make food only if embryo is sure to develop. The complex process of double fertilization was discovered by Russian biologist Navashin in 1898. It was first explained in Fritillaria and Lilium flowers. Let us have a look at the major changes in flower during sexual reproduction. In my previous video, I discussed with you the pre-fertilization changes. In this video, we have discussed fertilization process. In another video, we will be discussing post-fertilization changes. Separate videos would be there on reproduction in lower plants and on pollination.